Hello students, today we are going to discuss the last chapter of 9th standard. The name of the chapter is Division for Growth and Reproduction. In this chapter, we are studying about the process of cell division. Cell division. Let it be a cell. The important parts of the cell is the nucleus and the cytoplasm. The cell grows, and after a particular period of growth, the cell attains maturity. Okay, so here it is the mature cell, The cell grows and the cell attains maturity. After attaining maturity, the cell will start to divide, and in this chapter, we are going to study about the total process involved in cell division. The stage in which the cell gets ready for division is termed as interphase. I repeat, interphase. The stage in which the cell gets ready for division is termed as interphase. We can classify cell division into two types of cell divisions are there. So, we can classify cell division into two. The cell division can be classified into two. The two types of cell division are, one is the mitosis and the other one is the meiosis. Mitosis and meiosis are the two types of cell division. So, let us discuss the properties of these two types of cell division. Mitosis is a type of cell division which takes place in normal cell or somatic cell. Okay, is a type of cell division which takes place in somatic cell, somatic cells. And mitosis is actually responsible for growth. Mitosis is responsible for growth. In other words, we can say mitosis is the type of cell division which helps in growth. So, mitosis is responsible for growth. Meiosis is another type of cell division which takes place only in reproductive cell. This meiosis takes place only in reproductive cell. In other words, we can say that meiosis is responsible for reproduction, or it helps in reproduction. It helps in reproduction. In this session, we are going to discuss about mitosis in detail. Mitosis. So, before undergoing cell division, the cell matures and the cell gets ready to divide, and that stage is termed as interphase. The first stage involved is the interphase. So, interphase is the phase of a cell in which the cell gets ready for cell division. So, let us discuss some of the changes taking place inside the cell during interphase, some of the changes taking place inside the cell during interphase. The first change, the number of cell organelles increases. We already studied about cell organelles in our 8th standard. A cell possesses so many types of cell organelles. So, the number of cell organelle increases. The second change, the size of the cell increases. The size of the cell increases. The third and very important point is that the genetic material duplicates. The genetic material duplicates. We already discussed about genetic material. You know, the genetic material is DNA. In human beings, the genetic material is DNA. So, DNA is actually passing from one generation to the next generation, and due to the passing of this genetic material, some traits or some behavior are passing from one generation to the next generation. So, these are some of the important changes taking place inside a cell during interphase. Interphase is the phase of the cell where the cell gets ready for cell division, and the major changes are the number of the cell organelle increases, the size of the cell increases, the genetic material duplicates. Okay. So, now the cell is ready for cell division, and this cell division possesses two stages. So, let me explain which are the two stages in mitosis. The first stage is termed as karyokinesis. Karyokinesis. Karyokinesis means the division of nucleus or the nuclear division. Nuclear division is termed as karyokinesis, and this karyokinesis, this nuclear division is followed by cytokinesis. The nuclear division is followed by cytokinesis. Cytokinesis means division of cytoplasm is termed as cytokinesis. It means the division of cytoplasm. Division of cytoplasm. Now, the cell get ready for cell division. The cell is going to undergo mitosis. Mitosis is a type of cell division which is taking place in normal cells or somatic cell. Okay. And the stage in which the cell get ready for cell division is termed as interphase. In interphase, there are several changes taking place inside the cell. Okay. So, the first stage of mitosis is karyokinesis. Karyokinesis means nuclear division, which is followed by cytokinesis. Cytokinesis means the division of cytoplasm. Now, let us discuss different stages involved in karyokinesis. So, I am going to explain the different stages involved in karyokinesis. So, the karyokinesis consists of 4 stages. The 4 stages are, one is the prophase, the second one is metaphase, the third stage is anaphase, and the last stage is the telophase. So, karyokinesis means division of nucleus, and this karyokinesis possesses 4 different stages. The 4 different stages are, one is the prophase, second one is the metaphase, third one is anaphase, last one is the telophase. So, let us discuss what all are the changes taking place during all these stages. Let us discuss about prophase. Prophase. Okay, let it be a mature cell. Okay, a mature cell consists of, this is the cytoplasm, this is the nuclear membrane, and inside this nuclear membrane you can see a worm like structure. Okay, this structure is called as the chromatin reticulum. Chromatin reticulum. So, let us discuss the changes taking place during prophase. The first change taking place during the prophase is that the nuclear membrane disappears. You can, in this diagram you can see the nuclear membrane. This nuclear membrane disappears during prophase. So, this will be the condition of the cell, and this chromatin reticulum condenses to form chromosomes. I repeat, this chromatin reticulum condenses to form chromosome. So, already the nuclear membranes disappear, only the chromatin reticulum condenses to form chromosome. So, this is the chromosome. Here in this diagram, the nuclear membrane is absent and this chromatin reticulum condenses to form chromosome. So, these are the changes that happens during the prophase. The next one, it is the metaphase. The next stage is the metaphase. So, this is the cell which already undergo the prophase, and in metaphase the chromosomes are aligned at the equatorial plane of the cell. In metaphase, the chromosomes are aligned at the equatorial plane of the cell, and also from the pole of the cell a fiber develops which holds one part of the chromosome. This fiber which is developed from the poles is termed as spindle fibers. Spindle fibers. Okay, and these spindle fibers are developed from the poles. Okay, so the spindle fibers also develop during the metaphase. So, the next stage is the anaphase. So, let us discuss what all are the changes taking place during anaphase. Let it be the cell. During anaphase, that spindle fibers developed during metaphase pulls the chromosomes to the poles. As a result, the chromosome separates, okay, the chromosome separates from the equatorial plane and it moves towards the pole, and this separated chromosomes are called as the daughter chromosomes. Now, the chromosomes are actually moving towards the pole, and this movement is actually due to the pulling of the spindle fibers towards the poles. Okay. So, the last stage involved in mitosis is called as the telophase. The last stage of karyokinesis is the telophase. So, let me explain the various process involved during telophase. Now, the chromosomes are there in the poles, these are the daughter chromosomes are there in the pole of the cell, and during telophase the nuclear membrane reappears, and this chromosome gets converted back into chromatin reticulum. These chromosomes get converted into chromatin reticulum, and now you can see two nucleuses are formed inside the same cell, and after that, after a span of time, the cytoplasm also will start to divide, and finally from one cell two daughter cells are formed, and hence the mitotic cell division is completed. I hope it is clear for you. Thank you.